Very good afternoon, my dear friends. Welcome to the session. Our today's eminent resource person, Dr. A. Madhuri Madam Garu, Assistant Professor, Department of Microbiology, one of the prestigious institution in Hyderabad, that is GDCW Begumpet. Madam, please welcome and start your session. And please, Madam, share your screen. I have given you.
Good afternoon, sir. Thank you, sir.
Madam, please welcome.
Yes, madam.
Yes, madam. Okay. Sorry for the delay. Some technical issues. Okay. Right. So today we are here to learn about economic importance of bacteria. Means we are trying to understand how important are bacteria, whether it is a positive effect or a negative effect. It can be anything, right? Both beneficial and harmful aspects we have to learn today. First, we are concentrating on beneficial aspect. First role the bacteria playing is increasing of soil fertility. Because as we know, bacteria helps in all the microorganisms. They help in decomposition of dead and decaying organic matter. So by decaying all these materials, they will form humus in the soil, and thus they increase the fertility of the soil. And apart from that, these bacteria, they also help in producing nitrogen compounds or fixing of nitrogen, which is helpful in formation of proteins, nucleic acids, vitamins, etc. In a cell, all these components are there. Okay, so for manufacture of these components, nitrogen is required, which can be taken from soil and which is produced by bacteria present in the soil. Certain bacteria, they play important role in nitrogen fixation, ammonification, nitrification, and thus help in increasing the fertility of soil. We have studied about carbon cycle, nitrogen cycle, etc. Right? So in the nitrogen cycle, different steps will be there. So all these steps will be carried out by bacteria. Next, nitrogen fixing bacteria. Have you all heard about rhizobium? From childhood days, from school days, you are learning about rhizobium and nitrogen fixation. So this nitrogen, which is present in the atmosphere, about 79% of the atmospheric gases will have this nitrogen. Okay, but plants are not able to use that nitrogen directly. As they are absorbing sunlight, they are not able to use this nitrogen directly. What is the reason? Because nitrogen is inert gas. Inert gas means it cannot be used. It is not reactive. Okay, what is the reason for inertness of the nitrogen? The presence of triple bond between two nitrogen atoms. So plants can absorb nitrogen only in the form of nitrates. Nitrogen fixing bacteria can fix the free nitrogen in the atmosphere to absorbable form of nitrates. Means the N2, N triple bond N is converted into nitrates by these bacteria, and these nitrates can be absorbed by the plants directly. And nitrogen fixing bacteria are of two types: symbiotic nitrogen fixation and asymbiotic nitrogen fixation. What do you mean by symbiosis? Sym means together, bio means life. Okay, so two organisms living together and performing some function. So this nitrogen fixing bacteria, they are establishing symbiotic relationship with leguminous plants, right? And fix free nitrogen. You have studied about nodule formation, nodules in the leguminous roots, right? So that nodules are formed due to the presence of this bacteria. So these bacteria, they are forming nitrates and they are increasing the soil fertility. And these bacteria, that is rhizobium, mostly they are gram-negative motile bacteria that can grow in symbiotic association with the plants, and both plants as well as bacteria are benefited. Bacteria is giving nitrates to the plant and plant is giving shelter as well as food to the bacteria. And infection of the roots with suitable species of the bacteria leads to formation of root nodules. As you know, in Leguminaceae family, many different plants are there. Okay, but it is not like all the plants can be infected by the same bacteria. No, each plant will have different species of bacteria that can infect the plant. Means there is some species specificity for the leguminous plant infection. Bacteria like Rhizobium leguminosarum, Bradyrhizobium japonicum, etc. These are different nitrogen fixing bacteria. So we have just now studied that there are two types. One is symbiotic and the other one is asymbiotic. Asymbiotic means there is no symbiosis. There is no staying together of the bacteria, or there is no mutual interaction between two organisms. Okay. So these asymbiotic bacteria or free living aerobic nitrogen fixing bacteria, they live independently in the soil and they fix nitrogen into absorbable forms. These bacteria are the chief suppliers of nitrogen in grasslands and other similar ecosystems that lack plants with nitrogen fixing symbiosis. Means that in the plants, nodules are not there or symbiotic association is not there. In such cases, these free living bacteria will release nitrogen in the absorbable form. The example is Acidobacter and Azospirillum. Next is free living aerobic nitrogen fixing bacteria. Examples are Clostridium and Desulfovibria. They also help in atmospheric nitrogen fixation. And nitrogen fixation by cyanobacteria. Cyanobacteria also called as blue-green algae. Examples are nostoc and anambina. They also fix nitrogen from the atmosphere by specialized structures called as heterocysts. And by fixing the nitrogen, they are helping in the increase of fertility of the soil. These heterocysts are specialized cells where nitrogenous enzyme can be protected. And coming to ammonification step, this species of Bacillus subtilis, Bacillus macerans, Proteus, etc. They degrade organic material of dead plants and animals to ammonia and increase the soil fertility. So in this nitrogen fixation, first is ammonification, next is nitrification. Nitrifying bacteria like Nitrosomonas and Nitrophocus, they oxidize ammonia to nitrites, and Nitrobacter species oxidize nitrites to nitrates and increase the fertility. So in chemistry, you might have studied the sequence of reactions. Okay, so NH3, ammonia means NH3 or NH4 plus is converted to nitrite, NO2, and then nitrite is converted into nitrate, NO3. Okay, that is the final product of the nitrogen fixation. Coming to the bacteria, that is Actinomycetes, filamentous bacteria, that is Actinomycetes. They are also helping in nitrogen fixation. Example is Frankia. These are forming nodules in the roots of non-leguminous plants. So first case we have discussed is nodule formation in the Leguminaceae plants. Now we are talking about root nodule formation in the non-Leguminaceae plants. Okay, now example is Frankia. Next is phosphate solubilizers. These bacteria, they degrade phosphorus and they convert insoluble form to soluble form. Phosphorus solubilizers, they are converting insoluble form into soluble form. Example, Bacillus polymixa, Bacillus megaterium, etc. Now, bacteria as biofertilizers. Biofertilizers. Bio means life. Fertilizers means the compounds which can increase the fertility of the soil. Fertilizers means they can increase the fertility of the soil. Bio means life, means these are some living components that can be used to increase the fertility of the soil. They can be called as biofertilizers. So now we are talking about bacteria as biofertilizers, means how can the bacteria can be used as biofertilizers. So cultures of strains of bacteria which when applied to seed or soil or colonize the rhizosphere or interior of the plants and stimulate plant growth by increasing the availability of nutrients, means these biofertilizers or these bacteria, when they are applied to the plant seeds or in particularly to the plants when they are growing, they can increase the growth of the plant. And how they are improving the growth by improving cycling of nutrients and increasing soil fertility both ways. And they also help in increasing porosity and water holding capacity of the soil and provide protection against drought. Drought means dry conditions. So these bacteria, they are helping in water retention. Water holding capacity of the soil is increased and they are giving to the plant. And they are also increasing the seed germination. Now, by these biofertilizers, crop yield can be increased by 20 to 30% when they are cheap, convenient, and eco-friendly. And these biofertilizers, they have more importance in agriculture. For example, rhizobium. So, previous slides we have discussed there is species specificity for the rhizobium. Here you can see Rhizobium leguminosarum can infect host, that is pea plants, whereas Rhizobium japonicum can infect soybean and Rhizobium fascioli can infect beans. Means this particular species can only infect that particular plant. That is called species specificity. And Azotobacter, they are not forming any symbiotic association, but they are helpful in providing nitrogen in the fields of paddy, maize, cotton, sugarcane, etc. Similarly, Azospirillum is helping in production of banana, soybeans, paddy, etc. Right. So here I want to give some example biofertilizer. We have in our lab we have isolated rhizobium from methi plants nodules, and then our students have done project work where they have grown that rhizobium in our lab, and we have again applied to the methi seeds. So during germination and this growth is more in this rhizobium applied plants or seeds. Seed germination is more and growth is also more in the same sample. And bacteria as decomposers. Since the starting, we are discussing like bacteria are decomposing this organic materials in the soil. So whatever bacteria they are helping in this decomposition, they are called as nature's scavengers. Nature's scavengers. Just imagine if there is no decomposition. We are putting the dead body into the earth, and suppose there is no degradation or decomposition. What will be the condition on the earth? We can't survive. We can't stay. We can't. The earth will stink, right? But these bacteria are so helpful. They are degrading everything, decomposing everything. Okay. So such bacteria are also called as nature scavengers, as well as they are also called as saprophytic bacteria. Right. They are decomposing dead plants and animals, whatever is in the soil. And during this process, the bacteria break down the complex organic molecules into simple ones and make it available to plants. So what they are doing, they are degrading the macromolecules, whatever present in the cell. In the cell, what all are present, ma? There are nucleic acids, there are proteins, there are amino acids, there are carbohydrates, there are lipids. Whatever are there, all the molecules will be broken down into carbon dioxide, hydrogen, oxygen, simple compounds. In this way, they help in cycling of nutrients. So in your like previous studies, you might have known, you might have studied about carbon cycle, nitrogen cycle, phosphorus cycle, etc., which are being operated in the environment. So for this operation, these microorganisms are playing an important role. Without microorganisms, we can't imagine that cycles to be run in the environment. Next, in food production, you know very well, in curd, what kind of bacteria will be present? Bacillus is present. Likewise, butter, cheese, all these compounds, all these foods, they cannot be formed without the help of bacteria. For example, milk is converted into curd, butter, cheese, etc. by the activity of bacteria, and for that we need starter culture. Whatever culture we want to inoculate into the milk or other products, that is called a starter culture. And lactic acid bacteria, lactobacillus casei, lactobacillus acidophilus, lactobacillus lactis, all these are present in the curd. They grow in the milk and ferment lactose sugar. In the milk, lactose sugar will be there. So that sugar is converted into lactic acid, which results in the coagulation. And then a protein coagulation, curd formation is due to coagulation of casein protein. These bacteria are also used in the production of butter, cheese, etc. Coming to the production of yogurt or curd, the starter culture used for yogurt are Lactobacillus bulgaricus and Streptococcus thermophilus. You have to remember these names. Okay, what culture is used there. And production of vinegar, acetic acid, you know, I think. So this vinegar is also done by bacteria, and ethyl alcohol is oxidized to acetic acid by acetic acid bacteria, Acetobacter acetate, Mechanobacter species, etc. So ethanol is oxidized to acetic acid. That is nothing but vinegar. And next concept is probiotics. Here, these probiotics are good for humans in intestinal health. Okay, so these are living microorganisms which we have to eat or which we have to consume so that they can enter into our body and they can maintain our gut health or intestinal health. The most commonly used probiotic bacteria are lactic acid bacillus and bifidobacterium. If you have any idea, small children, when they have some problem, doctor will suggest some white powder. The name, trade name is some flora and bifidium, something of that kind. So if you see that powder, that is nothing but bacteria, probiotics. Next is fermented foods. Again, here, idli, dosha, all these foods are fermented foods. So in that also, we have to make the dough and keep it for overnight so that fermentation can happen, by which the microorganisms will make the content present in the dough to a softer this thing, and then we can get good dosha and idli. So this fermentation will also increase the taste or aroma and even nutrient composition of the foods. Next examples of fermented foods are kimchi, sauerkraut, kefir, wine. All these are fermented foods. Just you have to remember the names of the organisms which are involved in this foods. Next is single cell protein. Single cell protein means this concept of giving microorganisms as protein source has developed when the world has faced protein deficiency, because production was less and the population was increasing. So scientists were looking for alternative source of protein. So then they come across this bacteria as a single cell protein, and they have succeeded in this. And the first commercial SCP, single cell protein, is trade name is gluten. And here Megalophilus megalotrophus was used, and it was grown in methanol, and they produced 72% protein content. And it was used as an animal feed. And cellulomonas alkaligens can also be used as SCP. Spirulina, best example for single cell protein, that is algae. Okay. Next, in production of antibiotics. This you might be having a very good idea. Antibiotics, generally whenever we feel ill, when we go to doctor, they will suggest antibiotics. Okay, so what are these antibiotics? Break the word. Anti means against, bio means life, means against life, means it is killing something, someone. What it is killing? When we are taking antibiotics, it is going into our body and it is killing the pathogen or the bacteria which is responsible for the disease. Okay, so that is antibiotic. And so from where we are getting these antibiotics, some bacteria are able to produce the antibiotics, such as bacitracin and polymyxin. They are produced by bacillus. Whereas other antibiotics, streptomycin, tetracycline, they are also are produced by actinomyces. Next is production of butanol and propionic acid. In industries, the solvents, they are produced by Clostridium aestobutylicum and Propionibacterium species. Different organisms are involved in production of different compounds at industrial level. And enzymes. This enzyme is very important, streptokinase. Okay. Now, nowadays we are coming across many cases where people are dying of heart attacks, and many people are getting blockages in the heart vessels. So this streptokinase, it will dissolve the blood clots. This enzyme is produced by bacteria, Streptococcus mutans and Streptococcus faecalis. Such an important function. If we give streptokinase, that will dissolve the blood clots in the vessels. Next, in bioremediation. Bioremediation is cleaning of environment. Okay. This Spirituellus natans, it is popularly known as sewage fungus. It helps in degradation of organic matter in the sewage. And pesticide degradation. We like now there is awareness, there is maybe reduced usage of pesticides, but if you go back 10 to 15 years back, their pesticide usage was much like chemical pesticides, NPK, nitrogen, phosphorus, potassium fertilizers were used in the farms, and they have degraded the soil quality to a greater extent. So that pesticides can be degraded again by the bacteria. Okay. Next, oil degrading microorganisms. Like petrol is exported from Arab countries. So they are exported through the sea route, ocean route. When there is an accident, the oil can be, during all these accidents, this oil will fall into the water bodies and they will form a layer, which will kill all the aquatic flora and fauna, whatever fishes are there, whatever other organisms are there, they will die due to suffocation because oxygen cannot dissolve into the water, right? Due to presence of oil layer. So such problems can be solved again by the bacteria called Pseudomonas, Marinobacter, Acinetobacter, etc. And here we have to mention the name of great scientist, Indian-born American scientist. His name is Ananda Mohan Chakrabarty. So he has developed one microorganism or bacteria that is called as superbug. The name is Pseudomonas putida. You have to remember this organism name. It is very much important for your entrance. So Pseudomonas putida, what he has done is he has put some multiplasmid, different types of plasmids. Each plasmid is having a particular function. Okay, in oil there will be different components, 4 to 5 components will be there. Each plasmid will have a gene to degrade one component. So all 4 to 5 plasmids are put into one bacteria, that is Pseudomonas putida, and he called it as superbug. The components present are camphor, xylene, octane, hexane, and naphthalene, toluene, etc. So each compound can be degraded by a separate gene. All the genes are present in separate plasmids, and these plasmids are put together into this superbug. Next, biocontrol agent. Biocontrol agent means, here you might have heard about BT cotton, right? BT there is Bacillus thuringiensis. Okay, BT brinjal is also there. That is also Bacillus thuringiensis. So why we are calling it as biocontrol means this living organism, that is Bacillus thuringiensis, is controlling the pest of that crop. If it is a cotton crop, there will be a pathogen for that or pest for that cotton crop, and this bacteria will kill that pest. Next, Agrobacterium mediated gene transfer technique. Agrobacterium tumefaciens. This bacteria can be used in genetic engineering experiments to transfer the genes. And production of biogas, methane, biofuel can be produced by using different bacteria. retting of fibers. Here, bacteria are involved in retting of fibers, means the fibers will be made soft. And leather industry also, bacteria are used to remove hairs, fat, and tissues. And biodegradable plastics, PHB, polyhydroxybutyrate. Nowadays, if you check the cesarean operations or any other operations, stitches will be put by this using this thread that is made of polyhydroxybutyrate. They no more use that normal thread. They use this PHB so that it can be degraded after 1 week. It will be naturally degraded in the body. Patient will not go back to hospital to remove the stitches. And uses of genetic engineering bacteria. E. coli is proficiently used in this experiments, genetic engineering experiments. And examples are Pseudomonas putida for clearing oil spills, Rhinococcus radiodurans. It is used as used for bioremediation to remove the nuclear waste, and humulin, human insulin, is produced by transferring the gene, and the human growth hormone somatotropin is used to treat dwarfism, means if the height is less, it can be increased by using this hormone, and production of biofuels, etc. BT crops, just now we have discussed, the Bacillus thuringiensis is will kill the pathogen. Harmful aspects, denitrifying bacteria. Like nitrifying bacteria or nitrogen fixing bacteria are important. There are other group of bacteria which is doing other opposite function. Means if this nitrogen fixing bacteria is converting into nitrogen to ammonia and then nitrate, but denitrifying bacteria, they will convert this nitrate back to N2. Okay, nitrogen, molecular nitrogen. Examples are Thiobacillus denitrificus. This is a harmful function.
Madam.
And bacterial diseases. Sir.
Madam, only 1 minute is left. Harmful effects will continue next class, madam.
Okay.
Bacterial diseases and plant diseases, animal and human diseases will discuss next class, madam.
Sure.
Thank you, madam. Thank you very much.
Okay.
Okay.
Thank you.
So this is a great day because of madam has a wonderful session, given a wonderful session because of without using of laptop, her phone using to share the screen and also explain very fluently. My dear friends, if you have any doubt or clarification, please do ask in the WhatsApp and also during the class hours. Okay, thank you. Madam, thank you very much. Tomorrow, same time, madam.